I am Jane Pickering, and I am the very newly arrived, 13 days in the job, Executive Director of the new Harvard Museums of Science and Culture. Thank you very much. So I'm not sure I've done anything to deserve any applause. I hope that maybe when I'm standing here in 5 years' time, you still feel moved to applaud. But anyway, we'll see. So I'm absolutely delighted to be here for many reasons. I was saying to Arkhat, our speaker this evening, this is my first lecture at Harvard. Up until now, I was at the Cue the Boos and Hisses at Yale up till 3 weeks ago, so where I spent 10 years as Deputy Director of the Yale Peabody Museum of Natural History. So I have to keep remembering to add the Yale. Now I'm here with the real Peabody. But before that, I was at MIT, so I'm a little familiar with the Cambridge sort of environment and everything. So I'm thrilled to be back here, and obviously thrilled to be introducing Professor Arkhat Abzhanov, who is giving our talk this evening, which I'll get onto in a minute. But I first wanted to just give you a couple of introductory things. First, of course, most important, please silence your cell phones. So I'll just say that straight up. But perhaps most importantly, this is the first installment of the museum's 5-part spring series called Evolution Matters. And the next in the series in 2 weeks from now is Harvard's curator of butterflies, Naomi Pierce, who is going to be talking about how we look for signs of evolution in butterflies and bees and bacteria. 3 Bs, bees, butterflies and bacteria. And all the talks can be found in our wonderful brochure, which I was supposed to have to wave around, and I realized just I haven't got one to wave around. Anyway, hopefully everyone saw the brochure. They're on the table to the right, so you can pick one up if you don't have one. And I'd also, at the same time, encourage you to give us your email address, if you haven't already, because that enables us to keep you up to date with all the events. And I'm sure, Brian, we don't sell your email address to anyone else, right? We're very, very ethical about that. Yes, okay. And there's also information on the right that tells you how to become a member of the museums, which obviously we would really help as that helps support our mission. So before I get onto my introductions, I'd also like to recognize who I haven't had the pleasure of meeting yet, but Dr. Herman and Dr. Joan Suit, who I understand will surely say hello sometime soon, but who support the Evolution Matters lecture series, for which we're very thankful. And for those of us like myself who've been in the business of evolution education for many, many years, I know just how important this type of event is and how we're constantly working to really help people understand, you know, that evolution is the explanation for the wonderful biodiversity on our planet. And really sort of help people understand that. So thank you very much for that. And also to say that tonight's talk, along with many others, will be posted on the museum's website. And so I encourage you to go look at those, because all the past talks, I understand, are also on there. So to turn to our speaker this evening, who is Assistant Professor in the Department of Organismic and Evolutionary Biology here at Harvard, and where he and his colleagues strive to understand craniofacial evolutionary development in the vertebrate animal world, which, as far as I can see, especially what he's talking tonight, is about beaks and development of how dinosaurs became or were the ancestors for modern birds. And Dr. Abzhanov received his undergraduate degree from the National University in Kazakhstan, and came to the US in the mid-1990s to continue his studies in Indiana University, where he received a PhD in molecular biology. He came to Harvard in 2000 as a postdoc in the genetics lab at Harvard Medical School, and after spending several years teaching developmental biology at the Harvard School of Dentistry, he eventually joined the OEB department here at Harvard, where he's currently associate professor. His research has generated much excitement, which I think we're all going to enjoy this evening, and indeed was his major study on beak development in Darwin's Galapagos finches was actually declared one of the top 15 evolutionary discoveries of the last decade by Nature magazine, so that's quite an accolade. And he's received numerous other distinctions and awards, including a Templeton Foundation grant and a multi-year award from the NIH, National Institutes of Health, to study mitochondrial RNA. So a man of many talents and parts, and I'm really looking forward to hearing true interdisciplinary research. And I'd like to welcome you up to the stage.
It's my pleasure to be here. Just want to make sure that you can hear me at the back. Great. It's a very diverse audience here, and presumably you are here because you like birds. Hopefully you're going to learn some new information about birds from my lecture. The title of the talk, that's actually a more complete title that I had to shorten a bit for the museum, "Oh Little Bird, I Wonder What Art Thou," is a rephrase from an old Irish song. Somebody wondering what a bird is. And I have two goals for my lecture today. One is I'd like to tell you interesting stories about bird evolution. And two, I'd like to use stories as an example, as an illustration to show you how our field, my field of called Evolutionary Developmental Biology, where we try to combine advances in molecular biology, developmental genetics, and evolutionary biology, how it actually works, the techniques we employ, or the kinds of methods that we use. I was interested in birds since I was about 7 or 8. I became fascinated with birds. I enjoyed looking for their nests, peeking into their nests to look at the eggs and fledglings, and watching birds. Birds are extremely fascinating creatures, and I hope that I'll be able to share this fascination with you tonight. Birds have been around humans for all of our history. You can find birds in this prehistoric cave art paintings from 50,000 years ago. You can find birds in human religions. You can find birds in human fairy tales, songs, paintings, computer games. They're really everywhere. They're part of our culture. But few people actually really take the time to think about what the birds represent, what they are, how unusual they are. And I took this particular bird as an example because of how unusual it is. It's a mere robin that around Boston is one of the most ubiquitous birds. Now they're away, but they'll be back soon. They're so common that you see one probably every day, but you probably don't even notice it. Yet it's actually one of the most unusual animals that you'll find on our planet, certainly among the vertebrates. They have a number of features which are not seen in any other animals, and they're very different from their own closest relatives, which are crocodilians. The crocodilians and alligators are actually birds' closest living relatives. And look at these animals. They're completely different. Here's a creature which is all covered with scales. It has 4 legs instead of 2. It doesn't have wings. Obviously, it has a very long tail and has huge snout with lots of teeth. And these animals are each other's relatives. How could it be? Well, if we actually look at the history of the birds, which includes the fossils, the remnants of the kind of more intermediate lineages, what you find here is shown in this genealogical tree that we call a phylogeny that shows related lineages. So all these animals shown here, from crocodilians to birds, are called a part of the group called Archosaura. Archosaura from Latin means ruling reptiles. So since their origin in the Triassic about 230 million years ago, Archosaura tended to be dominant. They tended to focus on large prey. They tended to dominate the landscapes. And this Archosaura group includes crocodiles, very basally. They retain a number of primitive features. Pterosaurs, these flying reptiles, the other vertebrates which took to the air. All the dinosaurs, plant-eating dinosaurs, raptors, these bipedal raptors, and then primitive birds like Archaeopteryx and related species, and then modern birds. Unfortunately, much of that diversity is now gone all these lineages are now extinct. So we have modern birds, and then we have the crocodilians. And with all the intermediate lineages gone, it is a very stark contrast between these archosaurs. Let's go back to our robin. So robin has those unusual features that I mentioned. I'm going to discuss a few of them just as an example. So one feature which is obvious is the feathers. So robin, just like every other bird, has feathers. And feathers are actually quite unique to birds. All birds have them, but nobody else has this structures. We know that feathers evolved from scales. They're actually highly fancy, highly unusual scales. And if you take a scale and you kind of branch it out so they're all these branches connected to each other, you can generate a downy feather, which will protect you from the cold. It's a very useful structure, more so than just a simple scale. Then if you actually grow a shaft and have these branches called barbs from it, you can generate a contour feather that will make your body aerodynamic. You can add additional branches called barbules, and all of a sudden you have a feather that can actually be used for flight or as a tail feather. So these are very interesting, very unusual and useful structures. We now know that feathers evolved well before the birds. This animal here is not a bird, even though it looks like one. It's actually a dinosaur. It's one of the bird-like dinosaurs, which is covered with feathers. All sorts of feathers, contour feathers, the tail feathers, these wing feathers. It even has this wonderful crest. And we know not only about the plumage, that this bird had a plumage based on the fossils, we even know about the pattern on this plumage, because enough is preserved that we can actually understand the pattern and even the color. We know that it had this wonderful reddish crest on its head. And what this bird really looks like to me as a bird watcher, it actually looks like one of my favorite birds that I used to watch back in Eurasia, and that's the hoopoe bird, which is, again, this wonderful crested bird. And perhaps it's not hard to imagine that this animal here, very bird-like, was using its crest also, just like the hoopoe birds do, to attract mates and to intimidate its competitors. In addition to feathers, robins, as all other birds, have wings. And of course, all birds have wings, and wings are highly unusual, and they allow them to do powered flight. Only a few groups like bats, pterosaurs, and birds actually took to the air and actually have this active powered flight. In birds, the wings are highly modified hands, highly modified forelimbs. And in fact, this is actually a very gradual process. If you look at the early reptile or even early archosaur, we see this normal 5-fingered hand. All the fingers are present, all of them look fairly similar to each other. In more basal dinosaurs, the thumb begins to change. And in this kind of, raptors, these bipedal raptors, relatives of birds, the thumb now has a special joint that allows it to swing. They can actually now handle small objects. They have this really nice, very agile hands with long fingers. And then it's these hands that became the wings of birds. The fingers fused. Now they can actually use this wrist to support their own weight using the feather flights, the wing feathers. Another feature that can be used to distinguish a bird from, for example, a crocodile is its big skull. So it actually has a very prominent feature on its head, and so this is a skull, which is highly unusual, and we're going to talk about its evolution at the end of my second half of my lecture. But this beak is actually a wonderful, wonderful tool. So if you look at this picture here, the nest which the robin built, picking up branches and grass, and then using mud to cement all this into this really tough and waterproof structure, all that was built using the beak. Then it's using this elongated beak to catch a worm, to pull it out from the ground, to carry it, to feed its chicks. The chicks are using their beaks to catch the worm and eat it. So the birds are using their beaks as this wonderful and multitask tool in a ways that is not actually accessible to other animals which don't have beaks. Birds are very diverse animals. You can appreciate it here. Birds are also very successful. There are 29 orders, this kind of major groups of birds, and it's about 10,000 species. It's actually the largest group of land vertebrates. There are 9,000 species of reptiles. There are about 7,000 species of amphibians. That's the largest group of, and again, very successful, very diverse of land vertebrates. And much of this diversity, much of the success is due to ability to fly and these different beaks, which are highly adaptive for different foods, catching small shrimp, opening fruit and nuts, catching animal prey. In our group, we are particularly curious about songbirds, a group of birds which number about 5,000. They're called perching birds. And 5,000 species, that's actually quite similar to the number of species of all mammals. So again, it's a very diverse and very successful group of birds. And they also have this highly adaptive beak shapes. For example, this Hawaiian honeycreeper is using its bill to get to the nectar in the flowers. And this bullfinch is using its deep bill to crack seeds and so forth. So all these beaks have different purpose. To understand this diversity, we want to start comparing closely related species. So we compare apples with apples before we start comparing herons and eagles. And the ideal group to start is a group like Darwin's finches, shown here. Again, it's a group of 15 species, all of which different from each other, all of which closely related to each other. It's the entire radiation, the entire diversity of these birds evolved in less than 1 million years in Galapagos Islands. They all evolved from the small grass seed eater called grass squid, and they evolved to become seed cracking birds like these ground finches or the cactus finches, which feed on cactus flowers. And there are some insect catchers here. There is a bird called vampire finch, which drinks seabirds' blood and so forth. Very interesting variation, beak shapes which allow them to survive on different types of foods, which is a big plus if you live on the islands with few food sources. So here you can appreciate the differences by looking at the large ground finch, which is using this very deep and very broad bill to crack extremely hard seeds. No other birds can touch them. And here's the cactus finch. It has this shallow and kind of pointed, elongated bill to penetrate into the cactus flowers and fruit to get to the nectar. So again, very different beaks, very highly adaptive. So here you can see them, the same species, ground finch and the cactus finch in their native habitats, feeding on the flowers. This is the flower bud of a cactus, or cracking those nuts. And there are actually quite a few biology textbooks which compare different types of beak shapes in Darwin's finches to different tools that we use for different purposes, for doing very different things. So these are, again, you can see this is an analogy which is actually quite striking. Darwin himself, they're called Darwin's finches because Darwin collected them and brought them to science with his crewmates on the Beagle. And he's actually here writing about these birds that the most curious fact is the perfect gradation in the size of the beaks in the different species of Geospiza. That's the Latin name for these Darwin's finches, from one as large as that of a hawfinch, to that of a chaffinch, and even to that of a warbler. And these are different types of songbirds in England. Seeing this gradation in diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds, in this archipelago, one species had been taken and modified for different ends. Again, this is written exactly 20 years before his other famous book on the origin of species. He's writing this in his travelogue, The Voyage of the Beagle. And in a brilliant insight, he's using these birds as an example of adaptive evolution by natural selection. Since then, Darwin's finches have been studied by generations of biologists. This work is a collaboration with Peter and Rosemary Grants, who are at Princeton. They're ecologists, they're evolutionary biologists who studied these birds since the early 1970s. And they actually watched the evolution happen. They were some of the first people who actually recorded evolution happening before their eyes. They would go to an island and they would measure all the birds on this island. They would measure the beak shapes, they would measure the beak depth, the beak length, and they would come back to this island year after year. They would measure these birds and their progeny for many years, for about 40 years, 30 years or so, 40 years. And they actually watched through this period of time. There were multiple El Niños and drought periods and other interesting climatic events, and they watched how the beak shapes evolved over time in different species in different directions. So it's, again, a story which is ongoing. The beak shapes, not only they evolved, and we see them in songbirds, they're continuing to evolve today. So how do we actually study something like this? Well, the Grants noticed that when the birds hatch, when the Darwin's finches hatch from an egg, they already have different types of beaks on them. The ground finches have these very deep bills, and the cactus finches have these elongated bills. We very quickly found out that if you actually go into the egg, these are embryos. These are still several days before hatching. They already have the right types of beaks on them. The cactus finch embryos have these longer bills, kind of these very slender bills. The ground finches are developing these very deep and very broad bills. So whatever is regulating these beak shapes developmentally, that happens well before they're even used. It happens before they encounter the environment. They're built that way genetically. And that's something that's a very interesting thing to understand. How is knowing development, how things change in development can help us understand evolution? Now that's actually a very big question, which interested biologists for a very long time. So here's a process of evolution where you take a single cell and then it evolves, a single animal cell, and evolves through a series of transformations into a modern bird. So it evolves to be this, what we call a chordate, something that has an internal skeleton, a little cord inside, into an animal which has 4 limbs and the head and the tail. Then it becomes bipedal, then it evolves feathers and a beak, and becomes a modern bird. Then now consider development. During embryonic development, something very similar happens, the same as well. So you begin with a single cell, then it develops into an embryo which has a little cord inside, little internal skeleton called notochord. Then it actually develops 2 pairs of limbs. It develops a head with eyes. It develops a tail. Then it develops feathers, beak, and it turns into this adult bird. Very complicated organism. So how this process is related? And what do we, in understanding this relationship, how can it help us to understand development and evolution? So that's basically what our field is about. We're trying to use advances in both sciences, in evolution biology and in developmental genetics, to learn more about both, to use developmental biology to understand about evolution. And evolution tells us a lot about how development can change as well. In fact, Charles Darwin himself was actually very curious about embryology. That's how development biology was called during his time, which was actually exploding around his time as well. So he writes here that in the eyes of most naturalists, the structure of an embryo is even more important for classification than that of an adult. For the embryo is the animal in its last modified state, and then so forth it reveals the structure of its progenitor. He also writes in the letter to Asa Gray, the Harvard biologist, that embryology is to me by far the strongest class of facts in favor of change of form. So he really thought that embryology would be a very powerful tool to understand evolution. And then one of the fathers of evo-devo, of modern evolutionary biology, Richard Goldschmidt, states that evolution is the result of heritable changes in development. Simply put, in order to make something different for evolution to work on, you have to make it different. You have to have some kind of mutation that alters the developmental process. You shape the animal differently, and then it can actually enter the evolutionary process. So in our field, which we call evo-devo for short, there are 3 major approaches, and you will see how we employ them. The first approach is to measure evolutionary change. So how, for example, the beak shapes change, which direction did it change, how they change, what changed. 2 is we need to identify probable developmental genetic causes. And then 3, we need to know, so once we have this correlation, here's a morphological change, here's a molecular change, now we want to link them using some kind of a experiment. So we want to show that these are actually linked causally with each other. So Darwin's finches, to understand Darwin's finches evolution, we then need to understand their embryos. We need to collect their eggs, and for that we have to go to Galapagos, which is actually a wonderful field site. For those of you who are struggling with geography, this is a group of islands right here in the Pacific coast of South America. These are Galapagos Islands here. We go there before the rainy season starts. We look for nests which the males built, then we start tracking those nests. That's how the field looks like, the field site looks like on the Galapagos at the beginning of the rainy season. Looking for nests is interesting because birds they're not afraid of humans. They know mammalian predators. Some nests are really easy to find. Other nests are in less agreeable positions, and they are almost always built on those very thorny cacti. So let's talk about how we study finches. So here's the genealogy, this phylogeny, and the way it works is that, imagine this is you, that's your distant ancestor, that's your cousin Mary, and that's your very athletic brother. So if you take this tree and put Darwin's finches on it, so here's that grass seed-eating grass squid ancestor, and this is the finches we're comparing, so the sharp-billed finch has this fairly primitive-looking bill. It's not particularly long, it's not particularly wide. It's also used to feed on seed grasses, so on grass seeds, just like this ancestral species. That's this very highly unusual beak to crack hard seeds, and that's the elongated cactus finch bill. So what we wanted to find interesting molecules which control development, which would be expressed, would show some kind of interesting change that would correlate with this morphological, with this kind of shape changes, big shape changes. And well, first thing we actually showed is that the shape changes which you see in Darwin's finches are not random, far from it. They actually follow, in order to transform one big shape to another, they actually follow a particular type of mathematical geometric transformations called affine transformations that allow you to take a square object and squeeze it or stretch it or make it oblique. So very, very mathematical set of actually changes that they follow. And then we found our first molecule called BMP4. And this molecule, which normally regulates bone development, is you can see here the protein for BMP4 is very highly accumulated. It's present in very high concentrations in the beaks. These are developing embryonic beaks from a large ground finch. So we dissect an embryo and look for the presence of this particular molecule, and we find, again, this very, very strong levels of BMP4 in the large ground finch embryo. Well, here's another molecule called calmodeling, or calm for short, that's expressed and is present at very high levels in the cactus finches. And that's interesting because we have now molecular correlation, so some kind of change in the molecular level that, and this correlates with morphological change. But the question is, are these molecular changes significant? What do they mean? Are they enough? To address this question, we use the other very heavily underappreciated bird, the chicken. So the chicken actually has been a subject of scientific research for a very long time. And the reason for this is because if you take an egg, if you make a hole in it, you can see the embryo developing in front of your eyes from the beginning to the end. It's a very beautiful system. And the first person who actually studied embryonic development in chickens was the Aristotle. 300 years BC. He was opening these eggs, looking at the embryos, writing his observations. These days, we're using the staging system to understand how the embryos develop, which stages we want to target. And the idea here was to use this chicken embryo and to mimic some of those molecular changes, basically resurrect this force, this finch modifications onto the chicken embryo. So again, this is on the window. There's an embryo right there, and you can go there. You can use specially engineered viruses that force production of particular genes, for example, in the developing beak. And if we do that for that calmodeling molecule, for cactus finch, the molecule which is expressed in longer bills, in chickens, the chicken bills grow longer by about 16%, about the same amount that the cactus finches grow their beaks. And if we mimic that bullfinch situation, if you take the other molecule, BMP4, which is present high levels in bullfinches, this is a normal chicken embryo. So that's how a normal chicken bill looks like. If we force the bullfinch change, then we actually get this bullfinch-looking bills. Very, very deep, very, very broad. Something that, again, chickens never normally had in their in development or evolution. Moreover, we can show that if you carefully control the amount of this bullfinch molecule, BMP4 that you put in, you actually produce more and more bullfinch morphology. So that control is actually very tight. So the correlation is actually very strong. So in other words, if you bring about that molecular change, that will automatically cause the beak shape change as well. But we're seeing something really interesting, and on the surface hard to understand. The structural genes and molecules, the molecules which build up our bodies, or these beaks in Darwin's finches, like bone molecules and muscle molecules, are pretty much the same. They haven't changed in all these finches. The molecules which regulate those molecules like BMP4, what we call regulatory molecules, are also pretty much the same in terms of sequence. Yet we managed to produce different types of beaks. So the analogy that I like to use is building houses, building various buildings, where you use the same building materials, bricks and wood and other materials, you use the same tools, yet you end up building very different looking houses. So it's really when, how you use these tools that makes the difference. This is where the blueprint is. So this is how, depending how you use BMP4, where you use it, when you use it, you actually can make an interesting difference in beak shape. So that's the take-home lesson from the Darwin story. And now I'm going to switch gears and talk about a large-scale evolution of birds. Because if you actually look at the bird, like the seagull here, as I mentioned earlier, its skull looks almost nothing like the skull of its relatives and actually its ancestors, the dinosaurs and the related crocodilians. So what actually happened during the evolution of the avian phase? So the avian phase changed a lot during its evolution. So here I colored two bones. The facial bone is in green, and this little tiny bone which forms at the tip of the nose, of the snout, and in the crocodilians in red. And you can see here that by the time you turn into a modern bird, the face, as shown by green bone, almost completely disappears. Whereas this tiny little bone becomes the beak. So this kind of face and this kind of skull is actually quite unique in the vertebrate world. On top of that, there is something interesting happening during bird evolution when we compare them to the dinosaurs. So here's one of those bipedal raptors called Silophysis, a theropod dinosaur which is related to birds. That's an adult. It has small eyes, elongated snout, lots of teeth, fairly small skull, fairly small brain. But that's not how it starts. If you actually look at the embryo, late embryo, just about to hatch or just hatched, of Silophysis dinosaur, it has much larger, proportionally, much larger brain and eyes, much shorter face, fewer teeth. So it looks very, very juvenile. That's how actually other dinosaurs begin their life too. And then as they grow up, their face becomes longer and their skull, this brain part becomes proportionally smaller. If you look at primitive birds, like Archaeopteryx, here's a very large Archaeopteryx. Here's another one which is much smaller in size, yet their skull shape and their snout shape remains about the same. And even more importantly, the overall skull and snout shape is very similar to that of the juvenile dinosaur. To address this interesting question, what's happening during avian and dinosaur evolution, we set up collaboration with Dr. Mark Norell. He is a curator of the paleontology at the American Museum of Natural History. This Mark Norell, that must be some kind of TV host on the left. And Dr. Mark Norell is actually very famous for his studies in dinosaurs. And these things on the table are eggs, not just usual eggs. Those are dinosaur eggs, and some of them have fossilized embryos inside. So if you want to understand what happened during evolution and development of dinosaurs, Mark is the person to talk to. So what we did, we took the skulls and we placed this so-called landmarks, these little dots on the digital images of the skulls on particular bones. And we put them in the same positions in all these different skulls. And we looked at fairly primitive dinosaurs, fairly advanced dinosaurs, right before, like those closely related to birds, primitive birds, and modern birds. And then asked the computer to tell us, to track these points, and to tell us what actually is happening with the skull. How is it transforming as it evolves into a bird skull? And here's a summary that shows what's happening. And this is the basal archosaur called Euparkeria. It has this big face. There's a brain, eyes. Dinosaurs and very advanced dinosaurs. So this is kind of how advanced theropods, the advanced bipedal raptors look like. Very long face, very tiny little this kind of tip bone here, small brain. And then the skull begins to change. It turns almost 90 degrees in this kind of morphological landscape. And then here's Archaeopteryx. The face begins to shrink. This bone begins to, dark gray bone begins to expand. The brain begins to expand. The eyes begin to expand. And then you arrive to this modern bird. This is an emu here. Chicken looks very similar too, where the face now completely collapsed, the light gray portion, almost gone. The dark gray portion, the beak, is now fully very prominent. The brain is very large, the eyes are very large. And very importantly, all the skulls in this red area are in the baby zone, because that's where you find dinosaur juveniles and embryos. They begin here and then they develop to get to this, destination here. So the birds are becoming more and more juvenile-like as they evolve into modern birds. It's something called pedomorphism, a condition where adult descendant looks more like the juvenile of its ancestor. And one other famous example is the axolotl, which many people keep in their aquariums. It's actually an adult which retained a number of larval features, like this external gills and long fin tail and so forth. So birds then are highly pedomorphic juvenile dinosaurs. They retain a number of juvenile characteristics as adults. Why did birds evolve a beak at all? Why do they have it? Well, one idea is that it probably has something to do with its hands. Remember I told you how the close relatives of birds, these theropod raptors, had very agile hands. You can see here one of this, I believe a Compsognathus dinosaur devouring a lizard. It has this thumb, it has this very long fingers. And that's what the birds lost. As the fingers became incorporated and fused, incorporated into the wing, they lost those fingers. So the beaks allow them to have that very precise tool. Both upper and lower beaks are actually movable, so they can use them as pincers to build nests, to pick up food, to attack prey, to defend themselves. A very important tool. So where are we going from here? So I'm just going to mention some of the future directions. We don't have dinosaurs, non-avian dinosaurs. Luckily, we do have crocodilians. Remember that tree? Crocodilians very close to the base and birds in the top. Crocodilians still around like the alligators. To understand how the archosaurs, perhaps dinosaurs, had developed their snouts, we can look at the developing embryos of the alligators that we collect in Louisiana. And here you can appreciate how little the chicken embryo changes as it becomes the adult, how much the alligator changes as it becomes the adult, and how similar, again, the birds are to their own embryos, but also to the embryos of other archosaurs, like the alligator. So we have those embryos. We can actually look at the molecules which shape the snout in crocodilians. We can compare those with the situation in birds and perhaps do some interesting experiments. So that's work that's ongoing, and please stay tuned for the results. I have to mention that this is a project which has its own risks. So to collect eggs from a nest, you have to, we collect this nest from Louisiana from some of this, from alligators, and there is sometimes a female nearby, and so you have to be a bit, have to be careful. So to begin to summarize, I showed you how this approach that we use in evo-devo works well for small-scale evolution. We studied beak shapes, how they changed. We identified interesting molecules which correlated with those changes in the beak shapes, like BMP4 and Cal modeling. And then we did functional experiments to show that if you mimic that molecular change, you actually get that morphological, that beak shape change as well. So you kind of link everything. And we're using the same approach now to understand the evolution of birds as a group. So we are comparing, so we looked at the changes in the skull shape that I showed you. We're now identifying interesting molecular changes by comparing alligator embryos and bird embryos, and we're hoping to do some interesting functional experiments down the road. And just to come back to this very first question that I asked, what is a bird? Birds have a number of interesting features that they acquired since they diverged from the alligators, from the crocodilians. But they acquired them gradually, step by step, and often piecemeal. So, for example, if you're curious about the Archaeopteryx, there are all these debates whether it's actually a bird. Well, it depends where you draw a line. So again, this was a very step-by-step, very gradual process. And there are many species now extinct that have those different features. To summarize, birds are amazing creatures. I hope I showed you some of the really interesting things about birds. Birds are highly unusual modern-day dinosaurs. They're derived from dinosaurs. They are a subgroup within the dinosaurs, and they're also very unusual. Birds continue to evolve and change to this day. Peter and Rosemary Grants watched how bird beaks evolved on those islands in Galapagos. We can appreciate and even measure evolutionary changes in these birds. And also importantly, we can actually determine precise mechanisms for such changes by understanding developmental genetics and embryology. I'd like to thank all members of my group, all of our field assistants, all of our collaborators, all of our funding sources, present and past, to make this work possible. I'd like to thank you for your attention, and I'll take any of your questions. Thank you. What a great question. And are there any paleontologists here? Because turtles, position of turtles is a big mystery. Some of the paleontologists think that turtles evolved are closely related to archosaurs outside, but somewhere nearby. And others believe that they're actually outside of all the other reptiles. So that debate is ongoing. They do have a beak, but that beak evolved completely independently. And actually, it looks very different because they don't have this great expansion of this premaxillary bone, this tip bone. They actually fuse the maxillary bones, the bones which we have on the sides. So their beak is for cutting purposes, and it actually works differently. It looks different, but yes. And in fact, beaks evolved multiple times in dinosaurs. Triceratops, in this museum, that huge skull shows you how one can get a different type of beak. But birds have a unique beak because the way how they build it, nobody else has it. Question?
Yeah, it was sort of a stupid question. You've probably heard the expression stairs to teeth. So how long does the potential, if you have a regulatory gene, how long does that potential for teeth remain with the bird genome?
This was not a stage question, because there is actually a mutant, a chicken mutant, which grows teeth. How it happens is still not completely understood. We actually have this project. We found the stock for this mutant in California, and the idea is to study it a bit closer to understand the nature of the mutation that allowed for the chicken to grow. They grow primary teeth, the kind of teeth which develop first, and they're very kind of apparently crocodilian style. So it's been about 75 million years since birds lost teeth, modern birds. Any other questions? I'd have to take up. Yeah.
Why did the alligator have 5 toes and not 3 or 7 or 8?
So in biology, there are 2 answers. One answer is evolutionary. Why, for example, the dinosaurs have 3, right? Well, actually, it's this 3. 3 fingers, and alligators have, unless we have 5. Why that happened evolutionarily is one question. What happens developmentally? I can tell you that. But evolutionarily is actually still not clear why some lineages, for example, in horses, they only have 1, right? They only have 1 kind of a digit they're running on. They actually lost all the other fingers. So I don't think it's completely understood why dinosaurs have fewer and birds, as a result, have fewer fingers. But yes, I hope some interesting additional discoveries will answer this question. Yes?
I would like to ask you to expand a little bit on these drawings that you said were found in caves 50,000 years ago. Was this the first known drawing of animals, and where were these caves?
So some of the first drawings of animals were found in southern Europe, for example, in France. There are some wonderful cave paintings which date about 40 to 50,000 years ago. They're probably some of the first paintings, even though there are lots of carvings, so-called petroglyphs of animals, which are probably even older than that are found, for example, in Australia. So these are, as far as I know, these are some of the oldest actually images of the animals. And they show mammoths and the tarpon horses and birds and humans too. Oh, sure. Yes, sorry.
The molecules that you're finding that modify the bone structure, TA, LM, and the other, are you finding them in the embryos, or in the actual animal after they're born, or both?
Yes, so good question. The question is whether these molecules like BMP4 and co-modeling, those two examples, are found in the embryos or also in the adults. We detected them in the embryos. Kind of in 2 words, how we do it, we go to the field, we look for those nests, and then we find the time when the female begins to lay eggs, and we collect the last one. It's usually the 3rd or the 4th egg. The female lays a replacement egg. We take this egg, take it to the incubator, develop it, and collect the embryo. So this, I showed the images from the embryo. These molecules are expressed right around the time when the beaks are actually developing.
Okay, and do they continue to create these molecules?
Oh yes, if you look at later stages, these molecules are still involved.
And these molecules are just created as a result of the DNA of these animals?
These molecules, presumably there's some kind of a mutation that changes regulation of this gene, so that these molecules are present in all species of birds. In fact, they're present in all the animals, certainly all the vertebrates, like fishes would have the same molecules. However, they don't use them the same way. So there was some kind of regulatory mutation that allowed these finches to produce this particular protein in a new place at a much different concentration, and that brings about change in beak shape. So remember this same bricks, same tools. So yeah, it's really all animals have it, but when you actually use it is what makes the difference. Yeah.
How is it the same molecule that causes beaks to develop in the curve as well as
I know people are studying that, but I haven't seen the answer. I don't think it's known. There is an interesting related story that I'll mention. There's another researcher who was studying fishes, cichlid fishes. Cichlid fishes are these fishes which live in the lakes and in Central Africa. They kind of, finches live on these islands in the middle of the Pacific, and these fishes live in these lakes in the middle of Africa. So they evolve very rapidly as well. And some of these fishes evolved very deep jaws to crush mollusks. And he showed, Craig Albertson is this researcher's name, he showed that the molecule which actually is employed in a kind of higher level, it's expressed in higher level, and actually makes these very deep jaws in the cichlid fishes is BMP4. So just like Darwin's, these ground finches are developing this very deep bill to crush seeds, fishes are employing the exact same molecule in a very similar way to produce, to get very similar result. Yep.
Is any research being done to look for the actual regulatory mutations?
That would be a holy grail of this study, to kind of get all the way to DNA level and show and find the mutation. As I mentioned, it's very likely to be a regulatory type mutation, because we actually looked at the BMP4 sequence itself. It's essentially the same across all the finches. So it was going to be a regulatory change. And now we actually have sequenced one genome really well, and 3 of the large ground finch, actually the one which has this amazing bill, and 2 other genomes. There are more genomes coming online, so hopefully that will help us to bioinformatically analyze this bird's genomes and maybe find that mutation. Yeah. There are actually 4 now, so 3 sequenced by us, and there's another one. There's a consortium which sequenced 38 species. There's a talk of sequencing tons more in the future because the cost of sequencing is now so much less. So yeah, and so that's coming online. Yeah.
Thank you. Okay. Like, I'm guessing you've heard of that, how Jack Horner and some other scientists are trying to reverse engineer chicken. I was wondering, like, what you think of, let's see, of all that, how much it will actually contribute to, if anything, to what we know about bird evolution.
Well, if you have a very particular hypothesis in mind, for example, I know his work, he's curious about the tail. So in birds, instead of having a long tail with lots of vertebrae, they fuse it into style, into a single bone essentially. Now, if you apply the same set of methods to the other side of the body, to the tail, and found molecules that are expressed differently, say in alligator embryos versus bird embryos, and did some interesting function experiments and showed that you can actually develop, and basically explain how pigostyle forms developmentally. That would be useful. So yes, it depends on the question that you're asking. Question back? So the question was whether this regulatory change in, let's say, BMP4 is epigenetic. In case of BMP4, we know it is not epigenetic. It's fully genetic. These differences appear during development. They're already present at hatching, so before the birds actually use their beaks. However, they are definitely animals in which much of the skull eventual shape forms post-birth, post-hatching. Like, for example, famously in mammals, much of the eventual skull morphology actually is determined by interaction of muscles and bones. So a famous example is hyenas. If you keep if a hyena lives in the wild and cracks zebra bones for lunch, it develops this huge bones, very big crest for attachment of the kind of biting muscles. And if you take a hyena and keep it in a zoo from young age, it's not going to have those structures. It will actually have a very rather pup-like skull. So yeah, so the answer is depends. In this, birds is highly genetic, but they are definite examples where other influences are important.
So you've upregulated these molecules from either small beakers and large beakers. Have you tried the reverse? Presumably there are like multiple ways to go from small beak to large beak, and is that the way they're using it?
Yeah, so I didn't show here, but when we upregulated BMP4, we also used another virus which blocks this molecule activity. And if you do that, you get the exact opposite result. So the beaks actually shrinks. It most importantly, it shrinks in terms of width and depth. So it kind of becomes a slender version of itself. Yes. So it works the other way too.
There's some debate whether Archaeopteryx is a bird or something else. Do you have a view?
So again, it's a little smudged, but here's the debate. And again, the answer is what you take as bird features. So this old argument, as if it looks like a duck and quacks like a duck, has its own complications today because what actually you take as a bird feature. With more and more fossils coming online, tons of there were actually a lot of birds living at the end of Cretaceous. They died out with other dinosaurs. Lots of birds which look like jays or crows with little teeth. So there are many. Here's another related slide. There are many lineages which have those features in different combinations. So drawing that line, what actually makes a bird is an interesting and highly debatable process. So there is your answer. Question in the back. We don't know because we're not allowed to hatch them. Our animal protocol calls for collecting these embryos before they hatch. So what I can tell you is that when you make the beak bigger, when you upregulate BMP4 and the beak grows bigger, it still fits the face. That is, the rest of the skull changes to accommodate it, which is actually a very important feature because on one hand, we can change the beak independently of the body, and which is great developmentally because we see it happening evolutionarily. Yet there is also integration. So you can change the beak, but there's different structures around the skull are flexibly connected so that they listen to each other. So that actually suggests that maybe other structures will, there will be some interesting compensatory changes, and it may work out, but we don't know for sure. Yeah? It is believed that Tyrannosauruses, I'm not sure about T-Rex specifically, they're actually somewhere above this line because as juveniles, they probably had something like downy feathers. So they had these big fluffy chicks, which is kind of cool. There's some relatively big dinos now shown to have different types of feathers on them. Yeah, so the feathers are actually being found further and further, deeper and deeper in the dinosaurs. We don't know because you have to have a fine imprint of an animal. For example, the Archaeopteryx, the reason why we know for sure that all those famous birds coming from China, why they had feathers is because they fell into this shallow lakes and they became part of the silt, and they they have these perfect imprints, sometimes even containing pigmentation in there, so you can see these outlines of the feathers really, really nicely. That's hard to do with a 20-ton animal. So that's probably.
Fluffy T-Rex, maybe.
Well, thank you so much.